my pleasure to introduce today's speaker, Dr. François Paquet-Durand, an internationally recognized expert in neurodegeneration and inherited retinal diseases. Dr. Paquet-Durand is an associate professor at the Institute for Ophthalmic Research at the University of Tübingen, Germany, where he leads the cell death mechanism group, and he played a key role in advancing translational research in retinal degeneration. Dr. Paquet-Durand received his training in biochemistry and neuroscience and completed his postdoc trainings in Germany and Sweden. His work has been instrumental in elucidating molecular and cellular mechanisms of neurodegeneration, with a particular focus on inherited retinal diseases. Over the years, he has successfully coordinated and led numerous major national and European Union-funded research projects, including FP7, Horizon 2020, and BMBF initiatives, reflecting his leadership in large research efforts. In addition to his academic contributions, he has actively translated scientific discoveries into therapeutic innovations as a co-founder and the CEO of Murica Medicine. Dr. Paquet-Durand has authored more than 100 highly cited publications, highlighting the broad impact of his work in the field. He is also a highly regarded reviewer of leading scientific journals and also internationally funding agencies. He is fluent at academic level in French, Spanish, German, and English, and he also knows Swedish. As for today's presentation, I bet it will be English. Okay, please join me to welcome Dr. François Paquet-Durand in his presentation developing PKG inhibitors for the mutation-independent treatment of retinitis pigmentosa.
Wow, this was an introduction. I don't think I ever got one as nice as this one. So thanks a lot for this. Shall I start? Okay, so I'll start with a disclosure slide, which basically says that, well, I have been involved in patents and in this company that was mentioned already, Murica Medicines, which I no longer serve as CEO, but I'm still a shareholder. Well, I've also worked as a consultant for Graybird Vision, a California-based company at the time. So basically what this says is that I do have, or I may have, a commercial interest in the things that I'm saying. So you should take everything from now on with a grain of salt and question whatever I say. And I'm looking forward to critical questions in the end. So as an overview to this presentation, I will be talking about protein kinase G as a drug target for retinitis pigmentosa. I will introduce very briefly the topic of inherited retinal degeneration and what cell death mechanisms may govern this, and then focus more on cyclic GMP signaling in retinitis pigmentosa and why PKG may be an interesting target. I'll then talk about the expression and where it is localized, or where it may be localized, and then what happens when you inhibit PKG. And then I will talk about the preclinical development of a new compound, which we identified a couple of years back, cyclic GMP analog, which we refer to as cyclic nucleotide 238 or CN238. And if I have time in the end, I will talk about, or try and talk about what we're envisioning in terms of clinical translation. Okay, so to the topic of inherited retinal degeneration, this cartoon on the left shows an idealized cross-section through a retina. We start with the choroidea, the endothelial cells, the Bruch's membrane, and then the RPE cells, which are linked by tight junctions. And this is important because it forms a part of the blood-retinal barrier. Then we have the photoreceptor outer and inner segments. And then we have the outer nuclear layer, which harbors the nuclei, the cell bodies of rod and cone photoreceptors. We have rods, which in the human situation are responsible for vision under dim light conditions, and we have cone photoreceptors, which for humans are responsible for high-resolution color vision. These photoreceptors, rods and cones, connect to second-order neurons, which are residing in the so-called inner nuclear layer, and these are bipolar cells, amacrine cells, horizontal cells. There's also Miller cells that span the whole retina. And then the second-order neurons connect to third-order neurons, the ganglion cells, the retinal ganglion cells, and the axons of these cells form the optic nerve, which transfers visual information to different centers in the brain. So when we have an inherited disease of the retina, this usually leads to degeneration of photoreceptors, and hence this outer nuclear layer shrinks. Most often the rod photoreceptors disappear first and the cone photoreceptors disappear second. Cones may stick around for quite a number of years in some cases, but they're not functioning from a relatively early stage onwards. So these diseases are, from a genetic point of view, very heterogeneous. We nowadays know about at least 300 different disease genes, and clinically these are grouped into about 25 different diseases. And to name just a few here, there's Leber's congenital amaurosis and retinitis pigmentosa, diseases which primarily affect rod photoreceptors. And then we also have diseases such as achromatopsia or Stargardt's, which primarily affect cone photoreceptors. In these rod diseases, retinitis pigmentosa, we have the rod photoreceptors carrying a genetic mutation, which leads to the demise of the rod photoreceptors. And then secondarily, for reasons that we don't really understand, the cone photoreceptors, they also die, and then eventually complete blindness sets in. So most of these diseases are essentially all of these diseases are untreatable. We would obviously like to try and change that. So what mechanisms may be underlying the degeneration and loss of photoreceptors? A variety of different processes have been mentioned in the literature, and most often you hear this term apoptosis. And in classical apoptosis, cell death of a cell, of a neuron perhaps, is triggered by intrinsic signaling, which leads to the upregulation of pro-apoptotic genes, their transcription, their translation into protein, and then the formation of the mitochondrial outer membrane permeabilization. So the outer membrane becomes leaky of the mitochondria. This leads to the leakage of the mitochondrial protein cytochrome C, which together with apoptotic protease activating factor 1 forms an octameric wheel-shaped complex and with caspase 9, a so-called initiator caspase. This leads to the activation of further caspases such as 3, 6, or 7, so-called executionary caspases, which degrade pretty much all the proteins of a cell and eventually promote cell death. Now, apoptosis is by far not the only process that could potentially govern photoreceptor degeneration. Another process that is, well, a candidate at least, is necroptosis, which traditionally is associated with extrinsic signaling, activating tumor necrosis factor receptor, and then tumor necrosis factor receptor-dependent proteins, including RIP kinases and the cascade of RIP kinases and multi-lineage kinases, leading to the formation of damage-associated molecular patterns, which are immunogenic, and then cells from the immune system, microglia, and perhaps macrophages come in and degrade the cells that highlight these molecular patterns. Another process is par-thanatos, usually triggered by genomic or metabolic stress or associated with these stressors, leading to damage of the DNA and the activation of a DNA damage sensor called poly-ADP ribose polymerase. And this forms poly-ADP ribose polymers, which are associated with the activation of a nuclear protein called mitochondrial nuclear protein called apoptosis-inducing factor, which leads to the activation of nuclear DNases and the fragmentation of the DNA, promoting cell death. On the other hand, PARP may also consume excessive levels of NAD, leading to a breakdown of energy production. And this is associated with massive influx of calcium and activation of calcium-dependent calpain-type proteases, which are kind of the counterparts to the caspases. They also degrade all the proteins in the cell. Now, what I'm interested in, really, is cyclic GMP-dependent cell death. And cyclic GMP is a second messenger, which you probably all have heard about, which activates on the one hand protein kinase G, leading to the phosphorylation of numerous target proteins. And on the other hand, it may activate the cyclic nucleotide-gated channel, which promotes sodium and in particular calcium influx. Now, this may trigger processes that we don't really understand, and they may lead on the one hand to excessive activation of histone deacetylases and also PARP, poly-ADP ribose polymerase, and also calpain-type proteases, which together are likely to promote cell death. So in the retina, we've been focusing on this process, and the evidence that cyclic GMP has something to do with photoreceptor degeneration is actually pretty old. The first paper that I'm aware of was published by Deborah Farber and Frank Lawley in Science in the early 1970s, where they found that cyclic GMP in the retina was increased in a mouse model for retinal degeneration, actually the first mouse model for retinal degeneration, the so-called RD1 mouse. And this cyclic GMP increase, when compared over a wild-type mouse, was quite important in the time frame from day 10 to 18 postnatal, which is the time frame in which most of the photoreceptor degeneration in this RD1 mouse model occurs. And a couple of years later, the same group showed that you can recreate the degeneration of photoreceptors artificially, and this was in frog retina, where they put a PDE inhibitor called IBM-X on this. And the degeneration of photoreceptors was dependent on the concentration of this PDE inhibitor, indicating that PDEs and PDEs hydrolyzing cyclic GMP might have something to do with the degeneration. Now, that was 1977. That was before the phototransduction cascade had been really elucidated. So it's quite remarkable that already at this early stage, cyclic GMP and PDE were, on the stage as factors important for retinal degeneration. So much later, we found that cyclic GMP accumulates in photoreceptors, not only in the retina as a whole, but in photoreceptors, in rod photoreceptors for the most part, in different animal models for retinal degeneration. And here, this is a cross-section through a wild-type control mouse retina, the outer nuclear layer, ONL, inner nuclear layer, INL. And to the right-hand side of this are cross-sections through mouse and rat retinas derived from different animal models, all suffering from mutations in different genes in PD6A, PD6B, C, peripherin 2, rhodopsin, and also here in a CNG channel gene. So in all these cases, we see an accumulation of cyclic GMP in the photoreceptor, indicating that this could be something of a unifying principle that might be, happening, if not always, but in many cases in inherited retinal degeneration. As I mentioned, retinal degeneration is, from a genetic standpoint, very heterogeneous, and this makes it also pretty difficult to treat, because if you have 300 disease genes, you might have to develop 300 different treatments for each of them. If you could find a common principle, such as cyclic GMP in this case, that would make the chances of developing a successful treatment much, much larger. So what does cyclic GMP do, or how does it work in the photoreceptor? So in the photoreceptor, cyclic GMP is produced by an enzyme called retinol guanylyl cyclase. Potentially, it could also be produced by other guanylyl cyclases, and a very interesting one is soluble guanylyl cyclase, which is activated by nitric oxide. However, we've been looking into this for quite some time, and we really do not find soluble monocyclase activated. So, expressed and activated in photoreceptors. So this does not play a role for photoreceptors and photoreceptor degeneration. The signal is terminated by phosphodiesterase 6, a photoreceptor-specific PDE. And then cyclic GMP can activate, as mentioned, protein kinase G, PKG. It could also regulate the activity of other PDEs, notably PDE 2 and 3, which are cyclic AMP PDEs. So cyclic GMP via PDE 2 or 3 might influence cyclic AMP levels. However, we have not found any evidence for PDE 2 and 3 being expressed in photoreceptors. Another potential target is the HCN channel, hyperpolarization cyclic nucleotide-gated channel. And another one is the cyclic nucleotide-gated channel, which is important for phototransduction in photoreceptors. So if cyclic GMP is kind of our, common principle that we're interested in for therapeutic purposes, which of these targets really is important for the degeneration? So let's look at the CNG channel first. A very interesting paper that came out in 1999 in Nature Medicine proposed that a specific blocker of the cyclic nucleotide-gated channel called d-cis-diltiazem would prevent cyclic GMP-dependent degeneration in the RD1 mouse model. The RD1 mouse model suffers from a PDE6 mutation, has high cyclic GMP. So d-cis-diltiazem was proposed to rescue that, or to at least delay that. And diltiazem is a registered drug for hypertension. I think it's on the market in the US since about the 1960s or so. So the idea that an available drug might prevent retinal degeneration was very, very interesting. Unfortunately, it turned out to be not true. There were a number of papers, many of which came out after this, couple of years later, not published in Nature Medicine, but they clearly showed that this would not work. And about 20 years later, we looked at this ourselves using two different enantiomers of diltiazem, L-cis and d-cis-diltiazem, which target the cyclic nucleotide-gated channel and also voltage-gated channels. And what we found was that inhibiting cyclic nucleotide-gated channels would not improve the degeneration phenotype in the RD1 mouse. In the contrary, it would actually increase photoreceptor cell death, and only photoreceptor cell death, not death of other neurons. So we think from this that the cyclic nucleotide-gated channel is not our target in this context. Okay, how about HCN channels? So we know that there's 4 different isoforms. HCN1 is expressed in photoreceptors, in particular in the inner segment of photoreceptors. And what you see here are ERG traces, the B wave of a wild-type animal and of an HCN1 knockout channel knockout animal. And you see that the return to baseline after initial light flash is strongly delayed in the HCN1 knockout animal, indicating that the HCN channel activity is important for returning to baseline and reducing the refractory period of a photoreceptor or of the inner retina. Another study looked at the degeneration effect of the HCN channel, and they looked at the progression of photoreceptor loss, here expressed as the thickness of the photoreceptor layer in a wild-type retina, and then in another mutant retina, the cyclic nucleotide-gated channel B1 subunit knockout, which has a slow progressing degeneration. And when they crossed that animal with an HCN1 knockout, creating a double knockout, then the degeneration actually progressed faster. So from that, we concluded that inhibiting the HCN channel would not be a good therapeutic strategy. So basically then we're left with PKG. So A couple of years ago, we collaborated with a company based in the Netherlands that had developed a kinome screening approach where they could screen hundreds of kinases at the same time. And we compared the resulting phosphorylation pattern between wild type and RD1 retina. And the kinase activities that they quantified here in this kinase tree looks as such. Red means upregulated kinase activity, and there's in particular PKG1 and PKG2, which is upregulated. And then they ran a relatively sophisticated bioinformatic analysis over the kinome activity data sets that they had created and came up with probability scores for which kinase was most likely to have caused the phosphorylation patterns that they observed. And here, calmodulin kinase 4 actually came out as first, something that we had in fact found a couple of years before. But a very high score is given to PKG1 and 2, indicating that PKG1 and 2 are likely to be contributors to the phosphorylation patterns observed, which in turn indicates that there's a high overactivation of PKG1, or that there may be a high overactivation of PKG1 or 2 in the disease situation. So it's an indication, but it's not a proof that PKG1 or 2 really drives the degeneration. It could potentially be other kinases, such as calmodulin kinase 4, perhaps. So what we then try to do is to look at the expression of PKG. And there were a number of older papers in the literature that looked at PKG gene expression. There's two PKG genes, 1 and 2, and PKG1 has two different isoforms. And when you look at this here, PKG1 and 2 genes, the antisense indicates that there may be expression in the inner segments and also in the internuclear layer. And it's kind of similar for PKG2, some in the inner segments, but apparently less. And you don't see that in the sense probes. A few years later, the group of Robert Feil did a similar PKG expression analysis using a LAC-Z staining. And they also found that PKG1 was strongly expressed in the outer nuclear layer and the photoreceptor layer, and also in the ganglion cell layer. And especially this photoreceptor staining was gone when they used the PKG1 knockout retina. So that indicates that PKG1 is expressed in photoreceptors, but which PKG1 isoform really is expressed in the rods? This is still not really clear today. We tried to get to grips with that by using RNA scope, a technique similar to in situ hybridization, but potentially more powerful, much more sensitive. What you see here is a cross-section of the retina stained for DAPI, where you see the nuclei. This is a conventional microscopy picture where the RNA probes are labeled in red. Negative control, and this is the PKG1 alpha probe, where you see relatively good signal in the middle of the internuclear layer. That might be Miller cells, perhaps. You also see some here and in the ganglion cell layer. For PKG1 beta, it is much clearer that it is expressed in the internuclear layer and probably also in some ganglion cells. And for PKG2, there seems to be strong expression in what might be horizontal cells. When we look at a close-up of this for PKG1 beta in the inner segments and PKG2, we actually find that there is a relatively good PKG1 beta signal, not 1 alpha. And there's also a little bit of PKG2 signal. And our interpretation of this is that this might be the rods and this might be cones. But this is not very hard evidence, I have to say. At any rate, we think that PKG1 beta is most likely to be our target in rod photoreceptors. Sorry, to switch on speakers. So the concept for our now developments is that we think that different gene mutations that cause inherited retinal degeneration lead to high cyclic GMP in photoreceptors. And under physiological conditions, that would activate the cyclic nucleotide-gated channel, which is required for normal vision. So partner in the phototransduction cascade. And if it's too much under pathophysiological conditions, it would also activate protein kinase G, and that leads to photoreceptor cell death. And we could actually show that by selective activation of PKG, we can cause photoreceptor death. So the idea for a therapeutic strategy would be to selectively inhibit PKG. And we think that cyclic GMP analogs are pretty good for that because we can tailor them such that they only inhibit PKG and not the CNG channels, so that they don't interfere with normal vision. And over the years, we have identified, we've looked at many, many compounds, but we have identified initially this one, which we call cyclic nucleotide number 3. And a couple of years later, this one, cyclic nucleotide 238, which is essentially the same structure. It only is different by one methyl group, which actually increases its potency by quite a bit. So this is our candidate, our drug candidate at this point. So how do we test this? And to explain that, I will briefly explain one of the methods that we very often use, organotypic retinal explant culture. We take the retina from a mouse eye, take it out of the eyeball, dissect it such that the RPE stays attached to the retina, then put it onto a membrane insert, put it into a 6-well plate, and then we can treat it with cyclic nucleotide analogs, for instance, incubate it and keep it in culture for several weeks. The culture conditions are such that the retinal pigment epithelial cells face the membrane, the culturing membrane. We can culture this for up to 6 weeks. Under completely controlled conditions, we have a defined medium, which we call R16. It is free of serum, free of antibiotics and antimycotics. And this completely defined conditions, we believe, is quite important for drug testing and assessing a drug effect. And so one initial study that I want to show here, it was done on the PDE6B mutant RD1 mouse. We derived the retina from this, put it in culture at postnatal day 5, keep it in culture for 2 days without any treatment to let it adapt to culture conditions. And then from postnatal day 7 to postnatal day 11, we treat it with cyclic GMP analog at a concentration of 50 micromolar. There's a medium change every 2 days. And then what we look at is cross-sections of the retina. Here is the untreated, non-treated RD1 postnatal day 11 retina. You see the outer nuclear layer on top, inner nuclear layer, ganglion cell layer. The nuclei are stained with DAPI as a nuclear counterstain, and dying cells are stained with the Tunel assay in red. So every red cell here is a dying cell. And then we treat with different cyclic nucleotides and evaluate, count how many dying cells there are. So this quantification results in plots looking like this. We here express this as the percentage of tunnel-positive cells in the outer nuclear layer. In wild-type retina, there's a certain base level of cell death that is due to the culturing conditions. And then in the non-treated RD1 retina, we have a relatively high cell death rate that is due to the mutation. And then with the different cyclic nucleotides, we see cell death either lower or higher. And anything that is below the RD1 level, we would interpret as protective. And if it's at the same level or above, then it's not protective. So here, these compounds number 3 and number 238 actually appear quite interesting. So the next thing that we do is we tested this in different models. And this is another PDE6B mutant, but this time the RD10 mouse mutant. The RD10 mouse degenerates more slowly compared to the RD1, which may make it a little bit more like, well, a little bit closer to the human situation, perhaps. But here we have a different culturing scheme because of the slower degeneration. We start the culture at postnatal day 12, let it sit for 2 days without treatment, and then from postnatal day 14 to 18, we treat it with cyclic nucleotide 238, and we look at cell death as evidenced by the Tunel assay as before, and then treat with different rising concentrations of the cyclic nucleotide, look at cell death, and then in parallel to this, look at the number of cone photoreceptors, which we label with a cone arrestin staining. And then this is all quantified. We quantify the number of tunnel positive cells in the outer nuclear layer. We quantify the number of ONL rows, so essentially the ONL thickness, but expressed as numbers of cells in one row. And we quantify the number of cones, and here we quantify them as a number per 100 micrometers of retinal circumference. Now, as you can see, the treatment at this relatively early stage in the RD10 degeneration does not result in any changes in outer nuclear layer rows. It also does not result in any changes in cones, so it's not toxic to cones, but it does reduce the cell death rate, the number of tunnel positive cells in the outer nuclear layer at concentrations from 50 micromolar upwards. So then we use this data set to determine the relative potency of this compound, calculating the effective concentration, the EC50, the concentration at which 50% of the maximal photoreceptor neuroprotection is achieved. And this is the same data set, just shown a little bit differently. And we can use this dose response curve to now calculate the EC50, which stands at around 9.3 micromolar with a relatively high variability, because we have relatively high variability in the data sets. And when you look at this, the effect may not seem to be very large, but you have to consider that this is in part due to the culturing conditions. And if you were to compare this with wild-type retinal cultures, this would be kind of the baseline level of wild-type cell death that you would get anyway because of the culturing conditions. So compared to that, the effect is actually not so bad. So then the question is, if this is protecting photoreceptors, does it also protect the function of the retina? And here we use a slightly different culturing scheme, because we want to have, the degeneration to progress longer. So we cultivate from and treat from P14 until P24 at a stage when most photoreceptors are lost normally. And then we measure using a microelectrode array setup. We measure what we call a microelectroretinogram. And here, this is exposed to a light flash, and then we measure the field potential of the retina at the respective microelectrode array electrode, and get curves like this. We use different compounds. Here, for instance, we use cyclic nucleotide 226, which in the previous testing was ineffective, and number 3 and number 238. And these are individual traces for individual electrodes on the microelectrode array. But because the array has, in this case, 56 electrodes, we can actually look at a larger area of the retina and draw what we call activity maps for these microelectroretinogram recordings. And in these activity maps, a blue color means no response, and a red color, or the redder it gets, the higher the response amplitude. So in the untreated situation at postnatal day 24, RD10 retina, we have basically no responses left. With compound 226, that doesn't really change. But with compound 03 and 238, we actually have an activity profile all over the retina. And we can quantify this as the photoreceptor responses, or the number of electrodes responding positively to a flash of light in the electroretinogram. And we can see that compound number 3, but especially compound number 238, lead to significant increase in photoreceptor and retinal responsiveness. So that's encouraging. Now, where do we go next? Now, we actually tested this same compound in a variety of different animal models to see whether it would be efficacious also in other mutants, in other disease genes. And what I want to show here is only one, and this is a new mouse model for a dominant rhodopsin mutation, the isoleucine 255 deletion, which causes a relatively rapid onset of degeneration, or a relatively rapid degeneration. So in this rhodopsin I255-del mouse, we start culture at postnatal day 12, let it sit for 2 days. Then from 14 to 24, we treat with the compound and see what happens. And we actually treated also with the compound number 3, not only with 238. And we ended the culture at different time points, in some case already at postnatal day 18, so around here, early on in the progression of degeneration, and at postnatal day 28, very late in the progression of degeneration. And as you can see here, when you just look at the DAPI signal, so the nuclear counterstain in the wild-type non-treated retina, P18 and 28, there's not much of a difference. But in the non-treated mutant, at P18, the degeneration has progressed. We have lost perhaps 30% of photoreceptors or so. But at P28, we have lost like 80% of the photoreceptors. We again use the tunnel assay to assess how many cells are dying at this stage in a non-treated situation, in a situation where we use compound number 3 or compound 238. And then we quantify this as the percentage of tunnel positive cells, wild type versus non-treated mutant versus mutant treated with compound 3 or 238. And then we quantify also the rows of outer nuclear layer cells, photoreceptor cells. Here we don't really see a difference at postnatal day 18. But when we look at postnatal day 28, we see a decrease in the number of tunnel positive cells by both compounds. And we actually see a slight increase in the number of ONL cell rows that are preserved by the treatment. And especially the treatment with 238, compound number 3 does not seem to be as good. So this is the outer nuclear layer harbors mostly rod photoreceptors, 97% in the native state normally, 95% perhaps. This tunnel measure and the loss of ONL rows, that probably relates mostly to rod photoreceptors. So how about the cones? So for this, we use cone arrestin staining, and this is shown here. Again, for 2 different culture durations, culture lasting until postnatal day 18 and a culture lasting until postnatal day 28 at the bottom. And we can now quantify the numbers of cones per 100 micrometers of retinal circumference. In a non-treated situation, this has regressed already at postnatal day 18. At postnatal day 28, the non-treated mutant has lost a significant number of cones. And in the treated situation, this loss is somewhat attenuated, especially when we look at postnatal day 28 by the compound 238. Now, at the same time, we look at the lengths of the inner and outer segments as they are stained with cone arrestin in this mutant situation. And we find that the lengths of these inner outer segments together is actually increased by the treatment. And this seems to be also significantly improved by compound 238. So we see that compound 238 has preserved a part of the retina. It has in particular preserved the cones. So does it also preserve the function? And here again, we use the micro-ERG technique, where we cultivate the retina until, in this case, P24 really. And we measure the field potentials under each electrode. The through line indicates the averaged measurements, and the dotted lines are individual measurements for the treated in red, untreated in black, and gray is also untreated, but not cultured, acutely explanted. Sorry, my headphones keep on switching off. Okay, so we can quantify this. We have the non-treated retina, these gray traces. This is non-treated and acutely explanted. So just an hour or so in the measurement. This is non-treated, explanted, and cultivated for 12 days until postnatal day 24, but not treated and treated. And we have a strong, a marked increase in the negative deflection of this micro-ERG, and this is highly significant. And again, because this is a microelectrode array, we have 56 electrodes in this case at our disposition, so we can look at the whole area. Blue again indicates no response to light, and the redder it gets, the stronger the response to light. So this then shows that we have a preservation, a functional preservation of photoreceptors also in a dominant model for retinitis pigmentosa, not only in a recessive model like the RD10, PD6B RD10 mouse, but also in the rhodopsin isoleucine 255 deletion model. Okay, fun fact, this is on the side. So when we looked at this, because we have a microelectrode array, and the original purpose of the microelectrode array was to look at ganglion cells, not so much at field potentials, we can also measure ganglion cell activity at the same time. And what we found was that in the non-treated cultures, after a flash of light, there was no activity of retinal ganglion cells, no spiking activity. But when we treated with compound 3 and 238, we did see spiking activity of the ganglion cells in response to light, and this inactive compound didn't do that. And we could also quantify that, and the effect was highly significant. So that suggested that the compound did not only have an effect on photoreceptors, but that it might also have an effect on ganglion cells. And the point about that is that here we're using explanted retina in vitro. And of course, if you take out the retina from the eyeball, you have to cut the optic nerve, to get it out of the eyeball. And when you cut the optic nerve, you cut the axons of the ganglion cells, and that causes a retrograde Valerian-style degeneration of the ganglion cells within a few days normally. But this is a culture. This is 12 days of culture, and normally we would have lost all ganglion cells and all ganglion cell activity, but here we still have activity. So that suggests that we also have ganglion cells, or at least more than we should have. And this is indeed what we found when we stained ganglion cells with a marker called RBPMS, which stains about 60% of ganglion cells normally. And in a non-treated situation, we very rarely found a positive cell, but in the treated situation, we actually found RBPMS positive ganglion cells. And also here we quantified this, and this was a strongly significant effect that was not mediated by the inactive compound. So that suggests that the compound could also have an effect on ganglion cell survival after you cut the optic nerve, after you cut the axons, after you perform a so-called axotomy. So that was interesting. So this brings me now to the conclusion, at least the first conclusion. So what I would like to summarize is that our target is likely PKG1 beta, which we think is expressed in rod photoreceptors. PKG2 may be expressed in cones. Inhibition of PKG protects photoreceptors in both recessive and dominant disease. And not only does it protect them, does it preserve them morphologically to some extent, but it also preserves the function of cone photoreceptors in recessive and dominant disease. So, and an additional serendipitous finding was that we also find a preservation of axotomized ganglion cells. So we think we might be able to turn this into a new treatment for retinal diseases, inherited retinal degeneration in particular. And of course, there are a whole lot of things to consider now. One is, of course, the underlying mechanisms. How does this protection, PKG dependent or inhibition dependent protection. How does this really work? We think that PKG, the negative effect may be related to aberrant activation of certain voltage-gated potassium channels, which potentially drive up the consumption of ATP and also the activity of the sodium-potassium exchanger. But at this point, this is really, is rather experimental at this point. Now, at any rate, independent of what mechanism exactly is behind PKG activity, if we want to develop a treatment, we need to think about a lot of other questions now. So for instance, how are we going to deliver our drug? If cyclic nucleotide 238 is our drug now, or drug candidate, how are we going to deliver it? What is the best route of administration? What are the pharmacokinetics of it? Is it toxic? Do we have a proof of concept in a large animal model, perhaps, not only in a mouse? If we want to go forward, we need to produce this compound according to the standards of good manufacturing practice. We need to set up the processes for that. And then eventually we need to engage in clinical testing. So I'm not quite sure how much time do I have left. I could go on for like four more slides.
Yeah, of course. Please go ahead.
Okay. Okay, so then I will spend the next 4 slides a little bit on this idea of developing a treatment. So first of all, how do we administer this? If you have a drug, of course, you could think about systemic administration, and we tested that. And what is our delivery system going to be? And initially, we looked at liposomes for this. So we tried different liposomal formulations, and the idea was that we could inject this intravenously and perhaps intravitreally, but we initially tested intravenous injection in rats. And here we assess the pharmacokinetic profile of this. If we, for the cyclic nucleotide number 3 initially, shown here in yellow, if we inject the free compound, the compound is rapidly excreted via the renal system, and the half-life of the compound in the bloodstream is only about 10 minutes. So that's really short. So it's unlikely to have a measurable effect, especially if you think about a chronic disease where you have to treat not for minutes or hours, but you have to treat for decades. So the liposomal formulation of this looks completely different. Here we have a retention in the bloodstream for much, much longer, and the half-life is about 24 hours in this case. So from that perspective, liposomes look like a good idea. And we tested that also in non-human primates, in macaque monkeys, after, on the left-hand side, an intravenous injection. And we got a similar profile. The free compound was rapidly excreted. Half-life was, again, on the order of 10 minutes. And the liposomal formulation in the macaque bloodstream was much, much longer. Again, it was not 24 hours, but it was a similar order of magnitude. After an intravitreal injection, we used 2 doses. The low dose was never detectable in the bloodstream, but the high dose was actually just above the detection limit of the method in the bloodstream. So when we injected into the vitreous body of the monkey eye, after a couple of hours, we could see a little bit of it leaking out into the bloodstream, but it was actually very low. Mind that this is a logarithmic scale. Okay, so initially we were thinking liposomes, that this might be the delivery system to use. And we worked quite a bit on that for several years, actually. More recently, we assessed how the free compound, this time again, compound number 3, would behave in rabbit vitreous in vivo. And here in vivo, in the vitreous, the half-life is actually something like 4 to 5 hours. So it's significantly longer than in the bloodstream, but of course, for a chronic drug administration, that's far too little. So how about liposomes? So we made liposomes with this compound, and this is approximately how they look like. They're about 100 nanometers in diameter. And then we can think about how would they release the compound over time. So we have two aspects that we want to cover with a drug delivery system. We want to deliver it to the retina, and we want to have it release over a long period of time. You have to imagine we have a chronic disease that progresses over decades in the human situation, and we want to administer it to the vitreous, and we don't want to have to do this like every other day because that's not going to be tolerated. We want to do this as few times per year as possible. So we need a delivery system that delivers it and that delivers it over a very long time. So now the in vitro release of different liposomes. So these are 18 colon 1 phosphatidylcholine based liposomes at different pH levels. pH doesn't have a very big influence on the release rate, a little bit perhaps. And when we look at that, is that at about 8 days in vitro, pretty much or most of the cargo is released. 80% or so of the cargo is released in 8 days. Now, again, we're thinking we want something that would release over at least a couple of months, not days. So this is not really useful. Now, can we change the liposomes to make them release over a longer period of time? And we tried a couple of modifications for this, using liposomes that had a double bond, that had no double bond, or that were 20 mers. And these 20 colon 1 liposomes, they actually released over about 18 days. Unfortunately, their membranes were also very rigid, making them not so easy to handle. So 18 days is better than 8 days, but it's still not good enough to what we really want to achieve. And another point now becomes a concern is, what is the therapeutic dose that we need to achieve in the eye? And just for the sake of the argument, we assume it may be something like 10 micromolar or above. And if we had different liposomes, mind that the colors here are different on one side and the other, but if we had different liposomes, so these 18 colon 0 liposomes, they release very fast. They release very fast, and they never actually reach above 10 micromolar in the eye. If we have the 18 colon 1 liposomes, they actually might reach over 10 micromolar, but they only do that for a period of time that's about 2 days long, so far too little. Now, if we have the slow releasing liposomes, which release over a longer time, that's nice, but the release rate is too low to ever reach above 10 micromolar. So the bottom line is, this is not working. And it is very, very unlikely that we can ever tweak liposomes to work such that we have a release over a very long time at a sufficient release rate. So after a couple of years, we actually abandoned liposomes and moved on to something else. And this is PLGA-based microparticles that were originally developed by the company called Graybud Vision, based in California. And these liposomes, these PLGA particles, they look like this. They're microparticles. This is a bit difficult to see here, but the average diameter of the big ones might be on the 40 micrometer range. And in vitro, they have a completely different release profile, because these PLGA-based microparticles carrying cyclic nucleotide 238, they don't release over a few days, they release over many days. In this case, we actually think that we can relatively easily achieve release times of 90 days, 3 months, or even longer. The key characteristics, their particle size are between 5 to 14 micromolar. Their polydispersity index is between 0.1 and 0.3, so it's relatively narrow. And their pH is between 6 to 7.5. And we can load relatively large amounts of drugs into these particles such that we get a relatively good drug load per milliliter of volume that we want to inject into the vitreous. So these PLGA-based microparticles, they look very interesting. And actually, Graybud Vision had already tested them in the clinic in 3 different clinical trials. So this would be a way to go. Now, if we want to develop this further, we need to now think about a lot of other things like chemical manufacturing control of the cyclic nucleotide 238, the active pharmaceutical ingredient, the excipients, PLGA, and these PLGA microparticles are actually pegylated, decorated with PEG, and we need to make this into a combined drug product, the compound plus the microparticle. We need to assess toxicity, pharmacokinetics, mutagenicity, and we want to go for proof of concept studies in large animal models, in this case, dog mutants, dogs that carry mutations in PD6A and PD6B. And eventually, we need to do pivotal toxicology studies that are required by the regulatory agencies for clinical studies, and that satisfy the standards of good laboratory practice. So the way this works is you initially produce your compounds to this, not to the standards of good manufacturing practice, API, excipients, and drug product. Then you test this in preclinical studies that do not yet conform to the standards of good laboratory practice. And if this looks good, then you use the data that you've gained here in the production process and process development to set up a GMP production, and then you start GLP studies. And if that all looks good, you may then afterwards enter into a clinical trial. So on the final slide, I want to have a little outlook into clinical development. So the first thing that you need to think about is the manufacturing, the GMP, good manufacturing practice production. And that usually requires that you have several batches of your compound produced, and they need to be reproducible. So the quality from one batch to another needs to be the same. Otherwise, you have to start again. And you need to assess the stability of this over long term and quality control the whole process. So the first batch that comes out of this GMP manufacturing, that could be used for toxicological assessments. You could assess biological activity, and you would have to set up biological activity assays that you run continuously so as to assess long-term stability of your product and of the different batches. And then you can use this batch to do a pivotal good laboratory practice toxicology study in an animal model or two animal models that are accepted by the regulatory agencies, usually a rodent and a non-rodent. And then in parallel to that, you need to think about recruiting patients for a clinical trial. And you want to assess these patients way ahead of your clinical trial. You want to know which stage of the degeneration they're in, how fast the degeneration is progressing. So you need to examine them over a couple of years. Now, when you have that, provided the GLP tox looks good, the manufacturing all looks good, you set up contacts with regulatory agencies, get ethical approvals and approvals for the clinical trial. And then you may have different trial designs. The one that I'm showing here has it that you have a phase 1/2A clinical study where you start with a low dose, then a higher dose, and the next higher dose. And eventually you select the dose that was the highest tolerated dose, and you go into a phase 2B trial where you have multiple dosing over a longer period of time. And this might be several years in some cases, or in our case, probably several years. And eventually you evaluate how this has worked. In parallel to all this, you should have a monitoring and data management, biostatistics, and pharmacovigilance in place. And this should ideally be performed by an independent entity such that you don't run into, well, weird problems with people finding things they want to find. Independent evaluation is the keyword here. So, of course, there are a number of open questions here. Patient recruitment is obviously a problem. It's a rare disease, so you don't have so many patients. Which genes and which mutations do you want to consider for a clinical trial? Here in Tübingen, we're in the lucky situation that we have one of the largest patient databases anywhere, with over 30,000 entries. So we can actually pick the gene and the gene mutation that seems to us most likely to show a benefit in the treatment. Although, well, whether that really works is then a question of trying. A question of concern or that we need to address in the near-term future is whether we are going for a single-dose escalation trial, which is what is shown here, single-dose escalation, or whether we go for multiple-dose escalation and a continuous treatment over several years just in one go. That would mean that we would do a phase 1, 2A, 2B trial all in one continuous go. And then the question is, for how long would we have to do this? Are we talking, how long would we have to do this to establish safety? Well, probably not so long. A year or so might be enough. How long do we have to do this to establish efficacy in a slow progression neurodegenerative condition? That is an open question. Conservative estimates put that number at 4 to 5 years. Or conservative, maybe that's still even optimistic estimates. So we would have to do a very long clinical trial to assess whether this is efficacious or not. And at some point, we may also have to think about compassionate use, provided that let's assume that the treatment works. How do we ensure that the people who got it during the trial will be able to continue the treatment as long as they need it, even if the trial has ended? Okay, so with this, I actually at the end, well, I have one more slide, and that's some general considerations about translation from basic research to the clinic. Initially, my own thinking, I was this person here. I thought, well, you have a nice idea, cyclic GMP, and well, you do some lab work and then you get a pill, and shouldn't that work? I was very naive. So actually, it is a lot more complicated than that. So you have a number of different phases, 3 different phases perhaps of research, translational research. First of all, you do a lot of basic research. You have to discover something. And I related to these early studies by Deborah Faber and Frank Lolly that were done 50 years ago, where cyclic GMP was first put into the picture here. You need to have in vitro test systems. You need to have in vivo disease models. You need to discover a lead structure, do proof of concept studies. And if that all looks good, then you should already think about protecting it, getting a patent on it. Even though you're not sure that this may ever really be translatable, you should already think about protection here, because if you do it later, it may be too late. Then you go into exploratory development, and now you need to think about completely different questions. Pharmaceutical development, delivery systems. Maybe you need to optimize your lead, so you need to talk to chemists about how to do that. You need to set up production processes to keep manufacturing under control. You need to do pharmacology studies, pharmacokinetic studies, toxicology studies. And if that all looks good, then you go into real development. And now you have to think about regulatory affairs, a question that I knew really nothing about only a few years back. And you need to think about, what's your business model? Is there a market for this? And what's the size of that market? And you need to think about how to do the clinical trials. And of course, if this is ever going to be a viable treatment that is going to be available for people in the long run, it needs to be also commercially viable, needs to be commercialized. So where do we stand at this point with this cyclic nucleotide number 238? So we've done discovery research, we're done with that. We've also protected the compound, the applications, the manufacturing process, and the delivery system. We're pretty much done with pharmaceutical development and lead optimization. The manufacturing is currently in progress. Also pharmacological studies and also studies in large animal models and dogs are in progress. Regulatory affairs, talks to regulatory agencies are in progress. Market evaluation, that was done already a couple of years ago and it was deemed to be viable. But of course, clinical trials and commercialization, that is still years away from us right now. Okay. Now,
it was a wonderful presentation. And we see your acknowledgement. We're a little bit over time, but we have many questions. So if you could just get to those questions quickly. So I have one or maybe three questions, but there were several also on the Zoom. One is that you have three PKGs. Is this compound non-specific to inhibits all of the three, or is there a preference against one of the isoform?
Now, the compound, the cyclic nucleotide 238, is specific. It inhibits PKG1 beta more strongly than 1 alpha and PKG2. It inhibits all three isoforms, but mostly PKG1 beta.
There were also studies, I think, or at least I have seen on some meetings, that cyclic GMP has a positive effect on glaucoma. Is that possibly through the same mechanism, you think?
I don't know about that. Cyclic GMP was for a long time highlighted as a neuroprotective signaling molecule. And especially when it came to synaptic stability and similar aspects, cyclic GMP signaling was deemed to be beneficial. I have doubts about that. But if you look into the older literature, especially the 1990s and early 2000s, you will find a lot of papers claiming exactly that. Whether this is true for glaucoma retinal ganglion cells, I really cannot say. I can only say that from our perspective, it looks more like too much of it might be detrimental. On the other hand, a little bit of cyclic GMP is probably beneficial.
Yeah, hi, François. This is Franz. Yeah, very impressive presentation. I had like one thing, as you mentioned in the beginning, that most of the degenerative diseases have this higher cyclic GMP, but there are also some mutations, specifically maybe in guanylate cyclase, that lead to reduced cyclic GMP. So I was wondering if you have thought about testing kind of specificity of this treatment, maybe as a negative control, if it works in those or not.
Well, it's a very good question. Yes, I would say that there are diseases, that belong to this inherited retinal disease spectrum that are that are likely to be not treatable with this approach. And you mentioned guanylyl cyclase loss of function mutations, where you have too little cyclic GMP, and that causes the degeneration. I would think that this is a disease where our approach would not work. But a couple of years ago, we made a review about what kind of mutations are likely to be associated with high cyclic GMP, hence amenable to this form of treatment. And we came to the conclusion that about 25 to 30% of all inherited retinal diseases or mutations in disease genes might be amenable to this treatment, which then still leaves out 70% or so where we don't know, and where at least a number of them will not be treatable.
Well, thank you for the presentation. This is Philip Kaiser in the room. In your kind of last slide there, you had kind of some check boxes of what you had done, and one of those was in vivo disease models. Now, unless I missed something, it looked like the data you showed us was really ex vivo.
Yes.
So my question, if that's the case, I just wonder if, you've got a kinase inhibitor, and I would wonder if you would want to first go to a true in vivo model and maybe do some additional target validation, for example, with like an siRNA against specifically that protein. And then you can really show proof of concept that, yes, that's the one that's really working, because, these are kinase inhibitors. You have a screen, I know you did that, and it maybe looks specific, but, who knows what's happening really in vivo. So thank you.
Yes, that's a very good point. So we actually did in vivo disease model testing. Some of it is published, but some of it is not published, and that didn't refer to it here. But you're right, because so we have done that, but we've actually done systemic treatments in vivo disease models and found it to be working relatively well. But we've also done local intravitreal injection treatments and found that to be working even better. So that's the way we want to go. And in terms of negative side effects that you might get from a systemic exposure, we really want to avoid even that from happening. So that's why we go for the intravitreal, the local administration route to limit the systemic exposure. Now, another point that you raised, all these different kinases, and how can we be sure that we don't, hit one that we don't want to hit? This is very interesting. We have about 700 different kinases in the human body, and most kinase inhibitors will actually target either the kinase site, the protein recognition site, or the ATP binding site. In our case, we have 3 kinases here that are unique because they have a cyclic GMP binding site. No other kinase out of the 700, the 697 others, they don't have a cyclic GMP binding site. Hence, we're pretty sure that we are very specific to the 3 PKG isoforms. And among the 3, which we have evaluated, we know that we hit PKG1 beta really well. I'm not sure whether that answers your question, but.
Yeah, pretty much. Thank you.
Hi, François. Can you hear it? This is Dorota. Wonderful presentation and really very, impressive development. I was wondering, and this might be a very naive question, and I'm sorry if you mentioned it, but what about cyclic AMP levels when you do your treatment? How does it affect anything about the other side of the aisle, the cyclic AMP?
So, to be honest, we haven't really assessed that. And for the most part, we didn't really have proper methodology to assess cyclic AMP in the photoreceptors itself, which would really be what we want. There are reports in the literature that indicate that in some forms of inherited retinal degeneration, there may be elevation of cyclic AMP. I think that's true for the RD2, peripherin 2 mouse model. But other than that, there's not much in the literature in the ways of cyclic AMP being involved in the degeneration. And so to cut it short, we haven't really looked at cyclic AMP.
Thank you. All right.
We have a couple questions from the Zoom. Pat Bacera, are you ready? Pat, are you still here? No. So how about Tiansen Li?
Yes. Hear me okay?
Yes.
Good.
Yes.
A very interesting theory. So the theory is high cyclic GMP underpins most forms of retinal degeneration. And if that is the case, one could make some predictions. And that is that if animal models are exposed to a constant dim level of light, it will be beneficial to the disease course as a sort of a treatment. But in fact, the opposite seems to be true. Most RP models are very sensitive to the environment of light. They benefit from being kept in constant darkness. And the only exception to this rule that I'm aware of is a mouse model with a dominant mutation GCAP1, which activates guanylate cyclase. So this mutation continuously activates guanylate cyclase, leading to very high cyclic GMP production under whether light or dark diabetic condition. It's no longer respond to calcium levels. And so your theory is indeed true in this particular model. And in this model, exposure to constant dim light actually show a therapeutic effect. So the theory that you propose seems to work very well in select models, but the difficulty is to generalize to other models. And I would like your comment. Is there evidence specifically can you sketch a biochemical pathway where peripherin or rhodopsin mutations lead to high cyclic GMP? Thank you.
Yeah. So first of all, I don't think that all disease genes, all disease gene mutations will be treatable. And for sure, we have to distinguish, for instance, in guanylyl cyclase, we have to distinguish loss of function mutations and gain of function mutations. You know, loss of function probably not treatable, gain of function may very well be treatable. And that goes for pretty much each and every disease gene. We have to look very carefully. Is it likely that this will lead to high cyclic GMP or not? Now, I'm not sure whether I got all your questions right, but I think you also referred to peripherin and rhodopsin. So maybe first, why would we have higher cyclic GMP in a peripherin mutation? And I can only speak for the RD2 mouse, which has a peripherin mutation that basically abolishes the formation of the outer segment. And we see high cyclic GMP in the photoreceptors, in rods and in cones in this situation. So where does that come from? How does that work? We're not sure. We're not sure. But the idea, or one idea, could be that the malformation of the outer segment leads to the ectopic expression of the phototransduction components, including guanylyl cyclase, PDE, CNG channel, and so on. And that especially the ectopic expression and dysregulated activity of guanylyl cyclase may lead to overproduction and accumulation of cyclic GMP, you know, where it shouldn't be in the rest of the cell. And for rhodopsin mutations, it may be somewhat similar. You may assume that in a rhodopsin mutant situation, in a loss of function mutation, you may not have an activation of the phototransduction cascade, not even minor activation of this. And then that brings me to the question of dim light, whether dim light would be protective. So I don't think there's any hard evidence for that. I know that in some mouse models, like the RD10, for instance, dark rearing seems to be protective. That is a PDE6B mutant, the RD10 mouse. In the PDE6B mutant RD1 mouse, dark rearing is not protective. So, and they have mutations in the same gene, but they're different mutations. So, the question whether dim light or dark rearing or perhaps brighter light would be protective, I cannot really address that. I think it may be very context dependent.
We had a wonderful presentation and great discussion. We could have a longer if the seminar would be shorter, but we have to stop here. And I wanted just to only make a very quick comments what Dorota asked. You should take a look on system pharmacology, where the GPCRs regulate cyclic AMP, and they have very profound effect on stabilization of the retina. So please join me to thank our speaker. It was really intriguing pharmacology today.
Thank you. Thank you.